Today, we're tackling one of the biggest mysteries out there: how to actually read our own genetic instruction manual. Okay, so I want you to imagine something. Picture the human genome as this enormous book, a book with 3 billion letters. Now, just one single typo in that entire book could be the difference between a healthy life and a devastating disease. So, with a text that massive, how on earth do you even start to look for those typos? I mean, that's the huge problem that has stumped geneticists for, well, for decades.
Which brings us to our first section, a library of unknowns. Because for the longest time, that's exactly what our genetic code has been, a giant library filled with mysteries. And get this, you, me, the average person, we're all walking around with more than 9,000 of these potential typos in our DNA. Seriously, they're called missense variants. And yeah, most of them are totally harmless, but some can be absolutely devastating. But here's the truly mind-blowing part, the real kicker. Until very recently, of all the possible missense variants out there, scientists had only figured out the effect of a tiny, tiny fraction. We're talking just 0.1%. The other 99.9%, a total mystery. They were just labeled variants of unknown significance. So that leads us to the next big question. What exactly is this one-letter typo we keep talking about? Let's break it down. Okay, so a missense variant is literally just a single letter swap in our DNA. Think of it like a recipe. This tiny change tells your cells to use a different ingredient, a different amino acid, when building a protein. Now, sometimes that swap doesn't really matter. It's like using a different brand of sugar. But other times, it can cause the whole protein to just, well, not work at all. Here's a great way to think about it. Imagine the sentence, "The cat sat on the mat." Simple, right? Now, let's just change one letter. The cat sat on the bat. See? The sentence structure is pretty much the same, but the meaning is totally different. And that's exactly what a missense variant can do to the instructions for our proteins. Okay, so we've got millions of these potential typos. The huge question is, how do we translate what they mean? How do we sort the harmless ones from the ones that cause disease? Well, this is where it gets really exciting. Scientists have just been handed what's basically an AI Rosetta Stone for our DNA. Let me introduce you to AlphaMissense. It's a brand new AI model from Google DeepMind, and it was designed to do one thing: tackle this exact problem head-on. It's an AI built specifically to read our genetic language and figure out what these single-letter changes actually do. And the scale of what this AI has done is it's staggering. AlphaMissense didn't just look at a few known variants. It analyzed all 71 million possible missense variants that can happen in our proteins. I'm not just talking about the ones we've seen before. I mean, every single potential one. And the results? They're incredible. Out of those 71 million possibilities, the AI managed to classify a whopping 89% of them. It predicted that about 32% are likely pathogenic, disease-causing, and 57% are likely benign, or totally harmless. All of a sudden, that massive, mysterious library has a detailed card catalog we can actually use. So you're probably wondering, how in the world does an AI even learn to read DNA? Well, the whole process is super complex, but the basic idea behind it is actually pretty cool and surprisingly elegant. Okay, so first, it's not starting from scratch. It builds on the incredible knowledge of AlphaFold. You might remember that's the AI that basically figured out the 3D shape of almost every protein known to science. Then it was trained on these huge databases of genetic info from humans and our closest relatives like primates. This taught it what a normal, healthy gene looks like. So by seeing which variations are super common and harmless, it gets really good at spotting the rare ones that are probably dangerous. In the end, it gives every single variant a score based on its potential to cause disease. And this brings us to the most important part, really, a new era for medicine. Because let's be clear, this isn't just some cool computer science project. This is a breakthrough that could genuinely signal a whole new chapter for medical research. And how do we know it's such a big deal? Well, it comes down to accuracy. When scientists tested it against the variants that we already knew about, the ones confirmed by human experts, AlphaMissense blew every other existing computer model out of the water. It wasn't even close. Let's just put this into perspective one more time, because this is the key takeaway. After decades of brilliant human-led research, we had managed to classify 0.1% of all possible variants. Then, in what feels like one fell swoop, AlphaMissense comes along and classifies 89%.
That is an absolutely insane acceleration of our knowledge.
So, what does this actually mean for scientists working in the lab? Well, for starters, it helps them prioritize which typos to study, which saves a ton of time and money. It's going to speed up the discovery of genes that cause diseases, and it could dramatically improve how we diagnose rare genetic disorders. And maybe the best part, the entire catalog, all this data, has been made available for free to the entire scientific community. It's a resource for everyone. And just to be clear, this isn't abstract science. We're talking about real-world diseases that are caused by these exact kinds of single-letter mutations. Diseases like cystic fibrosis and sickle cell anemia. By finally understanding the full landscape of these variants, we're taking a huge step closer to better diagnoses and maybe one day, even new treatments. You know, for decades, it felt like we were wandering through this huge genetic library where almost all the books were written in a language we just couldn't understand. AlphaMissense has finally given us the translation key. So the question we're all left with now is a really hopeful one. With the library finally open for everyone to read, what amazing cures are we going to find inside?